Hi, everybody. I am Professor Omar Al-Habib from College of Science, Nawroz, University, Duhok, Iraq. Going to present the lecture entitled The Effect of Iron Nanoparticles on some Biological and Hematological Parameters in Albino rats. With respect to the nanoparticles, they are a very small, tiny particles measured on nanoscales. Normally, their external dimension ranging from 1 to 100 nanometer. Nowadays, nanoparticles are widely used in many fields, such as industrial, biomedical, and agricultural field. With respect to the biomedical, it can be used in diagnostic imaging tool, drug delivery system, vaccines, and immunotherapy. With respect to the industrial fields, they have been used as catalyst in the preparation of sensors and photovoltaic devices. Also, it is used in agriculture, in food safety, and in cosmetics. Due to this extensive use of nanoparticles, we can see that they are occupying a great area in global research activity. With respect to the formation of nanoparticles, they are present or naturally or incidental, or it may be manufactured as a material containing these tiny particles. With respect to the state of the nanoparticles, they are either unbound in an unbound state or an aggregate or agglomerate states. Example of that, we have nanofilms, which representing one dimension, nanofibers and nanotubes, 2 dimensions, and nanoparticles, 3 dimensions nanoparticles. Due to the extensive use and increasing production of nanoparticles, due to the high demand to these tiny particles and rapidly proliferating field of nanomedicine, you can see that this led to the exposure of living organisms to these nanoparticles via different routes. Therefore, it's of great importance or great demand to evaluate the toxic effect of iron oxide nanoparticles associated with their uses in multiple applications. Thus, the main aim of the current study was, number one, to study the effect of nanoparticles, different sizes and doses of nanoparticles on performance, food conversion rate, and mortality. And the second part included the study of these nanoparticles on hematological parameters in albino rats, ratus nervigicus. With respect to the materials and methods, to evaluate the effect of different sizes and doses of iron oxide nanoparticle on performance, food conversion rate, and toxicity, 5 groups of animals were used. Each of them consists of 8 to 10 rats. To perform the whole experiments, 40 to 50 rats, male albino rats were prepared of 8 to 10 weeks old and weighing 150 plus minus 10 grams body weight. The rats were obtained actually from the animal house at the Department of Biology, University of Zakho. The animals were kept in polypropylene cages, dimensions 30 by 25 by 17 centimeters, acclimated to the laboratory conditions. The animals were with free access to water, standard rat pellet at libido. With respect to the environmental condition of the rearing room, actually set at 24 degrees centigrade plus minus 2, relative humidity 50 plus minus 10%, and photo period of 12 hours light-dark cycles. The rats were randomly separated or divided into 5 groups, namely the control, then we have AA, AB, BA, and BB. This table actually showing each group, the size and the dose taken. In addition to that, the control group. In control group, there is no addition of nanoparticles. In group AA, nanoparticles of 25 nanometer external dimension and at a concentration of 1 milligram per kg feed. With respect to the second group, AB, 25 and 10 milligram dose. With respect to the BA, 50 nanometer external dimension, dose 1 milligram per kg. And the last group, BB, also the size 50 nanometer and the dose 10 milligram per kg. With respect to the performance of the living rat, the following parameters were determined: growth rate %, feed conversion ratio or rate, and mortality rate. The second part included the measurement of hematological parameters, including RBC, HB, HTC, MCV, MCH, MCHC, RBC distribution width, platelet count, MPV, white blood cell, lymphocyte, and granulocytes. With respect to the statistical analysis, was performed using GraphPad Prism 8, and the results were expressed as mean plus minus standard error of mean. Statistical differences were determined by Donut test for multiple comparison. And after finding out the p-value by using analysis of variance, the p-value less than 0.05 was considered as statistically significant. With respect to the result, you can see that the effect of different doses and sizes of nanoparticle on the performance of the rat showed that the percent of body weight gain in iron-treated rats were not influenced or not changed. Only the change was mildly and insignificant. P was greater than 0.5 as compared with the control. In addition to that, also during the experimental period of 4 weeks, the animals were like healthy, showed no behavioral change or any sign of toxicity. This is actually representing the growth rate in different groups, and you can see they are more or less very close to the control one, and statistically there were no significant differences between any of the experimental group as compared with the control. With respect to the feed conversion rate or ratio, you can see they showed better, the nanoparticle treated groups, they showed better feed conversion ratio as compared with the control, since there were no significant differences between any of the groups studied with that of the control. Also, no death was recorded during the experimental period of 4 weeks, and all treated groups remained survival in spite of their treatment with iron oxide nanoparticles. This is actually the feed conversion ratio. Again, as we pointed out, there were no any significant differences between them. You can see that the control one, the black one, and the other, they are the experimental one. You can see that slight elevation, mild elevation or increase in the conversion rate, but this increase was not significant from statistical point of view. With respect to the hematological parameters, you observe that all hematological parameters showed mild and non-significant increase in most parameters in all treated groups as compared with the control, except the red blood cell distribution width, which was just significant at the level of 0.5 as compared with the control. So this is actually, these are all the parameters we studied. The values were more or less similar, except for the red blood cell width. You can see that P is 0.03. It means that it's less than 0.05, which is the only significant case. The remaining, they showed actually non-significant differences as compared with that of the control. White blood cell and differential count. With respect to the effect of nanoparticles on white blood cell and differential count as they are indicated in the table, we can see that iron nanoparticle in all treated groups except AB, a group slightly and non-significantly reduced the total white blood cell count. This means they were reduced in AA, BA, and BB group. The other group, AB group, which is 25/10, the white blood cell count was non-significantly increased. With respect to the platelets were non-significantly increased in their number in all treated groups as compared to the control. Whereas the platelet distribution width and mean platelet volume and large platelet concentration ratio, all these, they showed non-significant decrease in their values as compared with the control group. Here we can see this is the platelet actually showing increase, but non-significant in the remaining parameters of the platelet parameters, that's distribution width and mean platelet volume and large platelet concentration ratio. In all these, we will see that they were decreased, but they were increased, but this increase was significantly non-significant. From the results of the current study, it can be concluded that treatment of rats with different doses and sizes of iron oxide nanoparticles for 4 weeks did not influence the performance, growth rate, and feed conversion rate in these 4 groups as compared with the control, as well as no sign of toxicity was observed. With respect to the hematological parameter, nanoparticles, they produce non-significant increase in RBC count, hemoglobin level, and hematocrit value, as well as both MCH and MCHC. Also, they were non-affected. Also, the total number of white blood cells was not affected, whereas the number of granulocytes was reduced to variable extent. Thank you very much for listening.